Okay, let's look at this enzyme cofactor now. So what's mean enzyme cofactor? So when I say the enzyme cofactor, the first thing you have to know that they are non-protein structure. Okay, so what they do, they bind to the protein, usually enzymes, so that they can enhance the protein biological activities, or increase the biological activity, or even activate the activity of the protein. So those are we call as an enzyme cofactor. Are you clear? Okay. So generally, I can use an analogy in this case, okay, or simulation in this case. An inactive enzyme without cofactor is known as apoenzyme, while the complete enzyme with the cofactor is known as holoenzyme. So you look at this diagram. This is the protein or enzyme. Okay, this is a protein or enzyme. These are the substrate, for example, to bind. Can you see that they can't bind? Can they bind to the inactive protein? Cannot. So what we do, we actually add something. When you add this cofactor, this something called cofactor, now you can see that the substrate can bind. So a lot of time in our body, a lot of enzymes, they are apoenzyme. Apoenzyme basically means that they have no function yet. Basically, they are inactive. When we need it, only then we activate it. So when we activate it, then they can carry out the catalysis process. Are you clear? Okay. So again, you do not need to know the name of apoenzyme or holoenzyme. Just name it as inactive and active. Okay. So when they are activated, or they are activated with the presence of the cofactor. But in fact, what is cofactor? Cofactor, actually, we can divide this cofactor into two different groups based on how they bind to the enzyme. If they bind tightly to the enzyme, then we call them as a prostatic group. Prostatic group basically means that they are non-protein structure, bind permanently, okay, tightly or permanently onto our enzyme. For example, if you look at this prostatic group, they are tightly bound, they are non-polypeptide unit required by biological function of some enzyme. So in this case, can you see that this is a catalyst? It's an enzyme. So can you see the non-protein structure? You can see alpha helix. Can you see that this enzyme got alpha helix? Okay, so this alpha helix, then you can see that within these regions, you have the heme group. So this is a catalyst. Are you clear? The catalyst with the active sites. So can you see that the heme group? So heme group is a prostatic group. First, why is prostatic group? So what you need to know prostatic group, two things. They are non-protein structure or non-polypeptide structure, tightly bound to the protein or enzyme. Okay, so for example, in this case, we do know that catalysts that require heme groups to functions, right? The enzyme. Okay, or we can actually divide our cofactor, okay, into another group where they are loosely bound. They loosely bind to the enzyme. Okay, so under this loosely bound group, we have coenzyme and activators. So what makes them different? Basically, one is to activate the enzyme. Another one actually is helping the enzyme. So two different, okay? And also coenzyme, most of them, they are organic. Coenzyme, they are organic. Activator, non-organic. Okay, so this is how we differentiate both of them. So what's coenzyme? If you look at this coenzyme, they are organic highlights. Most of them, they are organic. Okay, molecules that are required by certain enzyme to carry out the catalysis process. So they bind to active site, they bind loosely, so not permanently, briefly they bind and participate in the catalysis, but we don't consider them as a substrate. This is very, very important. They are known as coenzyme, helping the enzyme, but they are not substrate. Okay, because our aim is not to change them. Okay, so students will be confused. Why they bind the active site? They are not the substrates. Now look at this. If I put my glucose, okay, and fructose into my active site or the enzyme, I will be able to make them, okay, can I see that, into sucrose. So in this case, both of them, they are substrate because the final product involve them, both of them. But coenzyme won't involve in the formation of the final products. So in this case, in this example, again, in this example, can you see that? I want to change the alcohol into the acetaldehyde. Can you see that? Alcohol to acetaldehyde. So involve the alcohol dehydrogenase enzyme. Okay, can you see that? So in this case, this what we call the NAD. NAD, you realize that NAD, they are not part of the aldehydes. The product is aldehyde. Can you see that? So it means that NAD is not the substrate, but we term them as a coenzyme. So what is the function of coenzyme? We're going to learn this one in A2. In A2, not seems like it's very far away, right? A2, but actually it will be next week. Okay, or the latest will be the following week when we look at the energy and respiration. Okay, so now what is the function? Their function actually, they act as an intermediate carrier. Carrier of what? Carrier of electron, specific atom, or even a functional group that are transferred in overall reaction. Okay, so look at this diagram. What they do now? Look at this. First, alcohol. We want to form acetaldehyde. We want to remove what? We want to remove the hydrogen atom. Are you clear? We want to remove the hydrogen atom. So if you want to remove the hydrogen atom, someone have to take it, correct or not? You cannot simply remove it and kick it out. So someone here is an NAD. So NAD, actually right now in the next topic, we're going to learn it NAD as the hydrogen atom carrier. Their function. So what we can see here is when this alcohol binds to the active site of the alcohol dehydrogenase, NAD, can you see that, also bind to it? Okay, so when NAD bind, NAD actually take the hydrogen atom and then release it as the acetaldehyde. So as I said that if you want to remove the hydrogen atom, someone have to take it, right? So NAD actually take the hydrogen atom. Okay, so write down, this one is called reduced NAD. Okay, NADH is termed as reduced NAD. So it's something like redox reactions. Okay, can you see that? There are coenzymes involved in the dehydrogenation. Dehydrogenation means remove hydrogen atom. But again, we cannot just remove the hydrogen atom. We must have someone to take it. So this someone is coenzyme, and the general role of coenzyme, they are intermediate carrier of electrons or specific atom, in this case hydrogen atom, or functional group. Okay, that's transfer in the overall reactions. So this is called as the coenzyme. Okay, so what you need to know for coenzyme, three things. First, they are organic molecules. So where coenzyme coming from? Coenzyme actually come, most of them come from vitamin B. Are you clear? Vitamin B. So when we take vitamin B, actually is to help us to form more coenzyme. Okay, so these coenzyme, they are organic molecules first because they come from vitamins, so definitely organics, and they bind the active site loosely, but they are not substrate. So they function as what? They function as intermediate carrier. Okay, for example, in this case, they are intermediate carrier for hydrogen atom. Okay, now the last one is an enzyme activator. Enzyme activator, basically, most of them, they are inorganic ion. They also bind loosely, but now, can you see where they bind? They bind to the allosteric sites of the enzyme. So what is the allosteric site? Active sites carry out the catalysis reaction. Allosteric sites actually is for other compound to bind, either activates the enzyme or inhibits the enzyme. We have two ways to do this. Activate means make the enzyme actually function. So you can see that in this diagram, active site is not complementary. The shape totally not complementary to the substrate, But when this allosteric site other than active site are bound with the allosteric activators, so this activator can be ions. So this activator, when it bind, you're going to change the shape of the active site, allow substrate to bind. So when substrate bind, then catalysis process can take place. Okay, so example here is the magnesium ions act as an activator to activate the hexokinase. And this enzyme, we're going to learn this enzyme next topics when you look at the respiration. A glycolysis process, what is the functions of the hexokinase? Okay,